welcome to tonight's webinar for EnGenic. My name is Tamara Talbot and I'm the Investor Relations Manager here at VentureCrowd. For those that don't know, VentureCrowd is a digital investment platform that specializes in alternative asset investments, specifically VentureCrowd and venture capital property development investment opportunities available to retail and wholesale investors. As of last month, we have over 63,000 registered members who have invested over 170 million with us. More than half of our investors are repeat investors, which means they have invested with us more than once, and we are very proud of that number as it brings solid opportunities to our crowd and it shows confidence in what we bring to the market. We also have a number of exits in our venture capital portfolio. Our first exit was J-Ride, which was listed on the ASX, generating 107% gross returns to our investors in under 18 months. Our second exit was the ASX listing of 4D Medical, which generated over just 300% gross returns for investors in under 2 years. The reason for that is that we are a purpose-driven business. Our mission is to find and fund the future together, a future that is good for founders, good for investors, and good for humanity. That means we only work with companies that have purpose at their core. And the way we assess that is ensuring that each company that we have on our platform meets one or more of the United Nations Sustainable Development Goals. Those Sustainable Development Goals include affordable and clean energy, quality education, and good health and well-being and increased technology. And that is exactly where EnGenic sits. EnGenic is currently running a pre-IPO capital raise via the VentureCrowd platform. What does this mean? Well, it's an opportunity to invest in one of the best biotech opportunities that is focused on cancer and viral pandemic solutions, which is quite apt with the environment that we're finding ourselves in currently. If you want to find out more information, please visit the VentureCrowd website and deal page at any time during tonight's session, and you can invest at any time during the fireside also. Now I'd like to introduce our panelists this evening. Co-hosting tonight, I have Alex Knight, the Investment Manager at VentureCrowd. Alex is a finance, corporate advisor, and fund manager with expertise in mergers, acquisitions, capital markets, and venture capital. As an experienced angel investor and venture capitalist, Alex invests in and partners with high-growth companies to give them the best chance of success. Alex is also joined by the joint CEOs and co-founders of EnGenic, Dr. Jennifer MacDiarmid, sorry, I do apologize for the pronunciation, and Dr. Himanshu Brahmbhatt. Dr. Jennifer is joint CEO and co-founder of EnGenic, who in 2016 was awarded the Westpac Australian Financial Review 100 Women of Influence Award in the innovation category. Jennifer is a member of the University of Technology, Sydney, UTS Facility of Bioengineering Industry Advisory Board, member of the ANU Business and Advisory Board, and a member of the Australian Centre for Nanomedicine's End User Advisory Committee. In a conjoint position as Associate Professor at UTS, Department of Bioengineering, she supervises postgraduate students and interns and is a strong advocate for young women developing careers in science and technology. Dr. Jennifer joins us with Dr. Himanshu. Dr. Himanshu has a strong background in R&D innovation and the biopharmaceuticals business, having successfully developed company operations in Australia, USA, and big pharma relationships spanning the globe. He is the principal author on several scientific and clinical research papers, including the high-impact publications in Cancer Cell, Nature Biotechnology, and Lancet Oncology. Dr. Himanshu graduated with an MSc from the MS University, Baroda, India, and PhD from University of Adelaide, Australia. He did his postdoctoral research at the Geneva Medical Center in Switzerland and the National Center for Research in Biotechnology in Germany. Dr. Himanshu has been awarded a number of national and international research grants, as well as appeared on several documentary films and television shows relating to EnGenic's technology innovations. Prior to founding EnGenic, he was the principal scientist at CSIRO. I'm going to, on that note, hand over to Alex and our two panelists. I do welcome all three of you and thank you so very much for your time. And to the investors joining us tonight, I hope you sit back, relax, and absorb all the amazing information that we're about to share with you on this amazing new tech. Over to you, Alex.
Thanks, Tam, for that great warm introduction. It's much appreciated. As Tam mentioned, we do have Dr. Himanshu and Dr. Jennifer joining us tonight. But before we introduce them and dive into EnGenic, I just wanted to talk a little bit about the healthcare and biotech landscape to really set the scene of tonight's discussion. So, as we all probably know, cancer today is really a leading cause of death worldwide, with over 10 million deaths in 2020 globally and 19 million new cancer cases reported in 2020. Beyond the unmeasurable impact that this disease has had on individuals and families, the global economic impact was calculated at 113 billion USD in 2016 and is expected to grow between 6 and 9% annually. Now, they're quite frightening statistics. In the United States, where most of the data is available, this figure was calculated to be 183 billion in 2015 and to grow to 246 billion by 2030, a 34% increase. The oncology market itself, and for companies like EnGenic, was valued at 136 billion in 2019. Now, it's forecasted to grow at a compound annual growth rate of 11.9% between 2020 and 2025. There's many statistics out there and insight forecasts into the industry, and it's expected to reach over 470 billion by 2026. Asia Pacific holds a big part of that growth, and we're big supporters of Australian technologies, particularly in the APAC region, but through the Americas, and that's why we're quite excited about EnGenic. With limited options for treatments in oncology, the demand for solutions is only growing. This is why we have confidence in EnGenic, and to tell you more about the company and their technology innovations, we do have Dr. Jennifer and Dr. Himanshu joining us tonight. Welcome, doctors.
Thank you very much, Alex.
Thanks for joining us tonight. So we're just going to have a bit of a conversation about EnGenic, technology, product, market, a few different things tonight. But if you wouldn't mind, could you just give us a quick overview of what EnGenic is and how it came about?
Yeah, so Jennifer and myself were researchers at CSIRO, but a couple of incidences had occurred at CSIRO where the CEO had come down with an incurable cancer, and a technician in my laboratory also came down with cancer. These people passed away fairly quickly, and it was a turning point in our lives because we were working on vaccines, and we felt that we could continue to tinker around with all these test tubes for the rest of our lives, and when somebody is in dire straits, we're just standing there helpless. We can't do anything. So, it was a big turning point, and Jennifer and I decided to really focus our attention on cancer. And that's when we came up with this little concept of a nanocell that literally ticked every box that one could imagine was the hurdle for cancer treatment. So it was a mere concept, and we took it to the CSIRO, but they said, look, we are not in human health, we are in animal health, and therefore, we can't support this project. So we decided to actually, from our top level jobs, literally step out, get out onto the streets and go and find money to set up the biotech company. That's how EnGenic came about. And from concept now, we are in the clinical stage, manufacturing, and now heading towards the commercial side of it, which is IPO and big pharma.
So the company is built around a little nanocell, Alex, that was originally derived from changing the way a bacteria divides. So this little nanocell comes off at the end of the bacteria, and we found that it could be loaded with any payload, drugs, nucleic acids, adjuvants, you name it. And on the surface, it has a sugar molecule onto which you can hang a targeting antibody. So we realized this was potentially very powerful for cancer because it could go directly to the cancer cell and not cause toxicity in other parts of the body.
Yeah, so you mentioned there about the nanocell, and you've developed a unique patent protected technology called the EnGenic Dream Vector, or the EDV for short. Can you give us a bit more insight into how that works and what the advantages of that technology actually are?
So we can purify that nanocell, the EDV, and we can load it up. So there are pores in the surface of the nanocell, and very simply, we can load it up with different payloads, like, for example, chemotherapy. And then when it's loaded up, we just wash off the excess drug. we put our antibody on the surface, and it can then, when we inject it into patients, it will run around in the body until it sees the leaky blood vessels that are always associated with a tumour. When a tumour grows, it provokes blood vessels to feed itself, and that's a very quick and aberrant way of blood vessel growth, and they always have holes in. So that the EDV is just the right size at 400 nanometers to fall out of those holes into the tumour microenvironment. And when it's sitting there, it then sees the tumour and the antibody attaches to the receptor, and it gets engulfed inside the cell. So no drug is spilt outside the cell. It goes in and it's broken down inside the cell, and the payload, the poisonous payload in this case of a drug, spills out into the cytoplasm, the cell, and it dies.
So you mentioned chemotherapy there, and I'll focus on oncology for just a minute. With what you just mentioned, what are the advantages of this technology compared to other oncology treatments that are available?
So that's a very interesting thing that you see. I mean, literally hundreds and hundreds of nanoparticles, antibodies, drugs, etc. All of that has been developed over almost 60, 70 years. And yet the problem of cancer seems to be almost like, these are incremental advances. We are still poisoning people. People still go through the horrific toxicities. Now, the biggest difference here is that this little nanocell appears to be a one-stop shop. As Jennifer said, it specifically, first of all, goes only into the cancer cell and drops the bomb inside the cancer cell. But above all, it does something truly amazing. Because it is derived from these bacterial cells, it alarms the body into thinking, oh my God, we've got bacteria. So what the body does, on the side, it starts to stimulate the immune system. That immune system now goes into the tumor environment, sees all these dying tumor cells, gobbles them up, and actually trains the body how to recognize cancer cells. So when the cancer burden was growing, the immune system was being shut down. In all cases, the cancer cell has to shut down the immune system, otherwise the immune system will go after it. But here, the shut down immune system is revived. It now goes and recognizes those cancer cells, recognizes what is unique about them, and in every patient where we've seen amazingly dramatic response, where person is literally weeks away from death and then suddenly everything disappears. And we say, how on earth can this happen? It can't be just killing cancer cells. What really happens is this immune system, once you wake it up correctly, once you train it how to recognize a cancer cell, even end-stage cancer patients, you can pull them out with no toxicity. Now, this is unbelievable stuff, which no other technology is able to achieve. So there are lots of synthetic nanoparticles, but they are inert. So they can't activate the immune system. There are antibody type of therapies, and there's CAR T therapy, where people are actually engineering a cell of the immune system to train it to go and fight this cancer. But there's just, sorry, that's just what's up. When I get excited, of course, I get coughing. I start talking over myself.
If it's any consolation, your passion is contagious. So
right. No, so these are the very, very big differentiators here, that no other technology is able to train the body to actually recognize a tumor. And we've now fully recognized that if you're ever going to get on top of this cancer situation, it is the body itself of every patient is capable of doing it. You train it and it does the job. And
so you mentioned,
yeah.
You mentioned some of the impactful results there. Can you give us a bit of insight into the stage of technology, and particularly where you're at in your clinical trials and the results you've had today?
Yes, so we have done lots of clinical trials. There's a couple of slides, Alex, that one is, it's got the mesothelioma patient on. It's about number 4, I think, in that little.
Yeah, I'll see if I can share my screen.
So we have done, let's see if he can. Yeah, so go down to the number 5, I think it is. That one. Yeah. So we've done a lot of safety trials, phase 1 clinical trials. And the one you see on the top is a mesothelioma trial where we used a little bit of DNA which gets lost in mesothelioma, so we replaced it. And we had remarkable response with overall survival, particularly in a few people. So about 15% of the people responded really well. And mesothelioma is a death sentence within 9 months, as you know. So this is just an example of one man. He got some publicity, so that's why we're allowed to show him. But we had about a 70% response rate in that, but the overall survival was only increased in a couple of people. So the same with this brain cancer. 1 person out of 8 had a remarkable response, and his paralysis resolved and his tumour shrank. So we look back at all these results, and we did some canine studies with dogs with brain tumours who came to a local vet, and they showed remarkable responses because they hadn't been treated with lots of chemo. When we looked back, we saw that there was something these people did that the people who didn't respond well had no evidence of, and that is a little chemical called interferon gamma, and it actually stimulates a whole class of immune cells called T cells. So we thought, how can we get everybody doing this? These guys had they responded already to the EDV, so that gave them a big increase in overall survival. How do we get other folks to do the same thing? So if you just go to the next slide that you've got sitting there. Yeah. We realized that we could make an adjuvant molecule, just a simple glycolipid, very easy molecule, and it would load up in the EDV, and in that way it triggers a different pathway and stimulates interferon gamma, and the interferon gamma then goes and gets all these good T cells and another group of cells called iNKT cells there, and they're very important in rushing into the tumour environment and gobbling up and firing bullets at the remaining cancer cells that have not been killed off by the chemo. So now our final product, which we're using in the clinic, is an EDV carrying a very super cytotoxic drug. We're using something we call it 682, because we're dealing with patients that are really late stage. So it's a super cytotoxic that can't be used normally, but wrapped up in our EDV, it's very safe. And we've added the adjuvant that will stimulate this different sort of immune response. And so we've gone into pancreatic patients. We've done about 17 patients, I think.
So phase 2.
Yes, and that's in Melbourne and Sydney, and they're late stage. They have run out of all treatment options. If you don't do the next one, but the one after that, please. I'm just putting this up as our results up to the 8th of September, which we've collected. COVID put a bit of a stop to us collecting from the hospitals. But if you see the top red, that's median overall survival, and that is everything in pancreatic cancer, because people on the right-hand side, you see they normally only live 10 to 12 weeks after they've been finished all their treatment that's available. We are getting an overall survival of 35 weeks, and that it's 3 to 100, 12 to 100 weeks, when you should be cashing in your chips at 10 weeks. So that is really dramatic. And the percent of people who have some survival benefit is 76%. Now, we put in an abstract to a big conference last year, even though it was virtual, and a big hospital in New York, which are very, they're in pancreatic cancer and other gastrointestinal cancers, and they rang us up after seeing it and said, this is amazing, we want to do a trial here in America. And the reason to go there is that we can go up the chain. Instead of having people who are almost nigh unto death, we can go behind the first line treatment. So pancreatic cancer, terrible, intractable tumour, but that's our bag. If we can make a difference in a tumour like that, then this will really put a stamp on oncology for the future.
Yeah, okay. So beyond oncology, you've got the potential to use it in other vulnerable diseases, infections, and future viral pandemics. What developments has been around COVID-19? How have you been using this towards COVID?
So COVID-19 is an interesting one where we realized that we could package various different types of molecules within these nanocells. So what we actually did was, first of all, once the pandemic started and the race for the vaccine started, and of course, AstraZeneca, Moderna, and Pfizer, those vaccines started getting used, a couple of things became glaringly obvious. One is, just like we've seen with other viruses like flu, every year the new mutant arises. And so every year CSL has to go and make a new batch of vaccine, and they have to take the mutant into consideration. And it was obvious to us that this mutant is going to become a bad issue because this virus is going to keep on mutating. And with the global, when you have even in UK 40,000 cases a day, I mean, that's a lot of mutant viruses cropping up on a daily basis. So we said, unless you address this mutation problem, we are still going to be faced with some serious issues on this pandemic. The second thing was that these vaccines had to be stored and transported at -20 to -70 degrees. And the half-life of it is only 3 to 6 months. So consequently, like when Israel said, oh, we are giving a donation to Australia, they only give the donation because in 5 days, the use-by date was gone. Right? So the half-life is too short. So and we knew that our EDV was extremely stable. So half-life was not going to be a problem. We also knew that the EDV we can store and transport at room temperature. For our American trials, we simply transport the whole thing at room temperature. Kenya, etc. Anywhere in the world you could send it. So storage, all of those issues were not going to be a problem. The key was, how are you going to address the mutant? And of course, Delta came along. And by that time, we had worked out two things. One is that our body, when it makes these antibodies against the virus, there are two different pathways. All these vaccines are making the antibody by a single pathway. And what that pathway does, it makes what is known as low affinity antibody. By low affinity, I mean it's a weaker antibody. That's one pathway. And that's a common pathway, even all our flu vaccines. They're making these antibodies via that particular pathway. But we also knew that in the body, there is another pathway. It's a much more convoluted pathway. But what that pathway does, it actually makes what is known as high affinity antibodies. These are like Velcro. They stick like glue. No matter how this virus mutates, these things can hang on. So we said that if you are going to defeat this thing, you are going to have to make high affinity antibodies. And so that's how we engineered our EDV. And lo and behold, when we did these animal experiments, we got amazing result. No matter which mutant you throw it at, the antibodies stuck like glue. And in the same experiments, we had other types of EDVs by which we could trigger off the conventional pathway. And you saw immediately the same sort of results that AstraZeneca and Pfizer is getting, that the antibodies are low affinity, they don't stick like glue. When the Delta mutant comes along, they fall off. So efficacy drops to 40%. So we thought this is absolutely incredible. We can utilize any of these treatments, but you have to go via the second pathway. So then we immediately approached the hospitals, and in Melbourne, we commenced a clinical trial. And what is very exciting at the moment, we've just analyzed the blood from the first two volunteers, and they are making fantastic antibodies. These are high affinity antibodies. We have tested it against all 4 mutants, and all of them neutralize greater than 90%.
These two guys are probably the best protected
in the country
against COVID.
So now the Sydney Hospital, Adventist Hospital, is going to start and join up the trial. and we're going to get more of the volunteers through. But once we clear 40 volunteers and show that, yes, indeed, look, here, this is high affinity antibodies, it can neutralize these mutants, nobody is going to go down, it'll be just like a flu. And that is, one of the last points there is that because we were dealing with cancer patients, we knew that the EDV, because of its bacterial origin, is bypassing this immunosuppressive pathway. So consequently, now when AstraZeneca and all these companies, they did their clinical trials, in the phase 3 trials, they excluded all the immunocompromised people. So cancer patients, HIV, hepatitis, organ transplant, all of those people were excluded. The reason why they've done that, because they already knew that these low affinity antibodies are not going to be able to protect people who are immunocompromised. And then they did the trials in recent last 8 months. They've done the trials in cancer patients, and yes, indeed, they're showing that the efficacy is less than 40%, which is as good as nothing. But we knew that via the EDVs in the cancer patients, we were triggering off that immune system in 3 hours. We're not even talking about 6 months. So consequently, we have also put an application at the hospital to commence a trial in cancer patients, and because that population is going to remain highly vulnerable. And the current vaccines are not going to be able to protect those people. So we are in a niche area. We are not going to say that we are competing with Pfizer. What we are saying is what you guys can't deal with, which is COVID vulnerable population, will come along and try and save these people.
So it really seems the key to this is the EDV and the technology. You know, you've shown that it can have various applications, whether it be late stage oncology or it be towards the novel coronavirus vaccine. Engenic has a pretty extensive patent portfolio around this technology. You know, you've got you're the sole owner of 435 granted patents. It's not under license. You actually own these patents, and you've got 67 more pending. Can you explain the advantage that this gives and how many of those are actually attributable to what you are developing?
Sure. So in fact, when we originally started the company, you know, people had said, look, you guys are just starting with an idea. By the time you finish preclinical work, toxicology work, clinical trial work, and by the time you get out to market, I mean, 20 years, you know, everybody knows that in cancer, you know, if you start from concept, by the time you get to market, a minimum 20 years is going to take. So people say, your patent life is going to run out. We said, no, it won't run out. And the reason for that is very simple. Because if you had a drug, if I have a drug and I say, okay, I'm going to patent this thing, 20 years later, people have modified the drug and, you know, your patent life is gone 20 years. However, when you come to cellular biology, a complex system like this, then you have families of patents that are intertwined. They're like a spider's web. So how to make EDVs from any bacterial source is a whole family. How to load drugs is another family. How to manufacture is another family. How to target them, another family. How to virus protect, another family. So, and now what happens is if somebody is smart enough and say breaks the purification patent, you still have to target it. You still have to drug load it. So they're all intertwined. It's a mesh. And when we are now filing our new patents, which is now already 20 years gone since we started the company, we filed a new patent, but we have to incorporate in entirety the old ones because you still need manufacturing. So that's why our patent life has now gone out to 2041. And the pediatric applications have not even been put in. So it will go out to 2050. Our patent attorney is in Washington, DC. He said, senior partner there, and he said recently, Himanshu, who of us is going to be alive when this patent expires? So it's one, I have never seen a patent portfolio across 50 years. This will be one of the longest patent lives ever.
It certainly seems that there's so many pieces in this patent portfolio which make up the key, I guess, jigsaw of the business. And let's talk about pieces of the business for a minute. So obviously there's more people involved in the business other than yourselves. Can you give us some insight to who is involved in the business and who the key drivers are behind the business?
Sure. So we have not just in Australia, but we have a subsidiary in the United States. So we have an office in New York. The chief financial officer is in New York office. head of, so VP of Business Development is also there. Clinical trials specialists in the United States, because of course we have clinical trials there. And since we are aiming to list on Nasdaq, all our Australian accounting had to be converted into US GAAP accounting. So therefore, we had to have a US-based CFO. We also have a chief operating officer in Australia who liaises between Australia and United States. And then, of course, we have the manufacturing vice president who takes care of the whole manufacturing, quality control, quality assurance. And we have a VP of clinical trials who's also here in Australia. He coordinates all the clinical trials that we have in progress. And so there's a core team of about 7 people, and each one has an area of specialization. Then we have the extended team, because to go on a Nasdaq listing, we need not just our Australian corporate lawyers, but we also need our US corporate lawyers, because like S1 filing and all that has to be done by US corporate lawyers. So for the last 6 years, a major firm in New York, which is Latham Watkins, senior attorneys there are our partners, taking all the way through to IPO. And additionally, our patent attorneys, Foley & Lardner is the law firm that handles the entire patent portfolio. They're based in Washington, DC. The chairperson of IP at the law firm is actually the head of our patent portfolio. And then comes the oncology team. Now, we deliberately shied away from one oncologist because this thing was going into so many different cancers that we realized very quickly that if you talk to a colorectal cancer person, then they don't know anything about ovarian cancer. So you had to have multiple specializations. So we currently have something like 30 oncologists in Australia and United States in different areas of specialization who are actually part of a core team. They advise on clinical trial protocols and all of these only in the cancer where we're dealing with. And lung cancer guy is completely separate. So the extended team is a very large team actually, but different areas of specialization.
Yeah, so it seems fair to say you've got a pretty healthy team behind you in very different areas that are going to support you through. Now, you did mention the Nasdaq IPO upcoming, and I do want to talk about that. But before we do, you've been through a couple of funding rounds previously, a Series A, a Series B, B1. Can you give us some insight into those capital raise rounds, who were involved, and what it allowed the company to do?
Sure. So, yeah.
So when we first started, it was death by 1,000 cuts, actually, because we were not known or anything, and we just had an idea. So we got seed money, and then we had to prove that we could make EDVs, and then we got a bit more money and such. So Series A really is a amalgamation of a lot of different groups coming together. We have had 2 Australian venture firms in there originally, and one of them is still with us because he keeps renewing. But the Series B came about because we were in the clinic and we saw the need to get some more strategic investment, especially from America. And so we have a couple of hedge funds from the US. I have to tell you that our patent attorney company and they, as individuals, have invested because they think this is out of the box technology. So that's a good tick for us. And Himanshu and I think often people don't expect CEOs to put money or founders to put money in, but we are very heavily committed, as best we can be, not being billionaires. We're very heavily committed into this company, and every time there's a fundraising, we put money in too.
My house has been mortgaged 3 times over now.
but when you've got skin in the game, you not only have the passion for the science, but you have a business interest in making sure that this thing gets out to people. And the only way it's going to get out to patients is if we have a healthy fundraising. So this Series C is, we're calling it Series C, but it's really a pre-IPO. When you get towards the Nasdaq IPO, you need crossover funds that are well known in the biotech world, and they come in sort of 2 or 3 months ahead of an IPO so they can gauge the pricing better. But right now we're raising 25 million to make sure that we have enough money to progress our clinical ambitions here and elsewhere, and that we can cover all our IP, which is very expensive exercise because our patents worldwide, PCT patents, and we're very grateful, frankly, for people like VentureCrowd, because you guys have, you've done it very quickly. We know where we are, and you've been very active, so that's great.
Yeah, well, we appreciate that. We like working with companies that we think are developing technologies that can really create a better future. And I do want to talk about the IPO market for venture-backed biotech companies for just a minute as well, because we've seen that particularly since COVID-19 showed its face in the global economy, we've seen that a lot of biotech companies have listed and raised record rounds, listing primarily on the US exchanges. The median biotech IPO size was around 451 million and was 34% year-on-year increase. So you're kind of well established in your engagement with most major US pharmaceutical companies and investment banks to make that happen. What would a potential IPO on the Nasdaq do for the business, and how will that grow the business?
So it's very interesting. So our strategy is a little bit different from many of the biotechs that have gone on the IPO on Nasdaq. So many of these biotechs have, in the IPO itself, they've raised in excess of 300 million to 500 million. Now, what that does to the shareholder base, it is a very serious dilution of people who were there pre-IPO, because you literally given away more than 50% of the company. What we have is we have such a strong news flow from all this interim data from so many different cancers. Then you'll have, breakthrough in pancreatic cancer. You got breakthrough in glioblastoma. You've got a pharma licensing deal. So all this news flow, pre-IPO and post-IPO, means that we should be able to keep that momentum going up post-IPO in terms of share price, which is very good for all shareholders. Now, we feel that in the IPO, 100 to 150 million raise should be plenty, because clinical trials can be done. That's where all the news flow comes from, pharma licensing deals, patent protection, and a bit in the manufacturing. But by that, when you push that valuation up, then in a year's time, if you wanted to raise more, which we don't think we will, because once you have a pharma licensing deal, each of these licensing deals come with 400 to 500 million upfront. So you don't need to go into even a follow-on. You can just have 2 or 3 licensing deals, and the company is well on its way. So strategically, it's very carefully thought through, and with the US banks working with us, they've been watching our news for everything, and so that's how the strategy has been set up. How to maximize shareholder value and not suddenly just give away most of the company to a major crossover type of fund in the United States, because then nobody else benefits. Whereas we want all shareholders to benefit. And one of the classic examples is BioNTech. Once they went IPO, their valuation was in the 400 million range. And the moment they struck the Pfizer deal, and by a stroke of luck, luck for them, not for the rest of the world, the pandemic came along. And in less than 2 years, they've gone from 400 million to 58 billion today. And they've had no follow-on raising.
Just keeping an eye on the time, and I can see a few questions backing up here as well, which we'll get to in a moment. But you did mention the Nasdaq IPO and the capital raise there. And I just wanted to give an overview for our audience tonight. So currently, we are running the pre-IPO or Series C Nasdaq raise for Engenic through the VentureCrowd platform. We initially took an allocation of 1 million, to which we've oversubscribed to 1.97 now currently in commitments. And you can find that on our our platform at any stage during tonight and obviously after as well. Tam, I'll just hand back over to you. There's a few questions that are coming in from the audience. Would you mind just running through some of those and we can chat about them?
Okay, some of the questions from the start of this webinar have been covered. So Lawrence has asked the question that has been addressed, but just for completion, I'm going to read out the questions. It's how many trials have been run out, how many people involved, and what were the results? So Lawrence, the recording of this webinar will be sent out to everybody. So for those that joined late, those questions will be addressed early on in this fireside chat. If you do need more information on that, please just send me a message and I'm happy to share that question. The second one is, if trials are successful, how long before therapy is available to the general public?
So very interesting. Okay, so point number one is trials, that 5 phase 1 trials, safety is okay. Okay, so the second one is the important thing to note is we have a high probability now of getting breakthrough status from FDA. So once you get breakthrough, after phase 2B trial, one can go straight onto the market. You still do phase 3 trial, but post phase, so you don't have to wait to phase 3 before you go on the market, because it is considered as a major unmet medical need. So after phase 2B, you've got breakthrough status, you go out. So consequently, one would say that if we get the breakthrough status by early next year, then in less than 2 years after that, the technology can get out into the cancer patients, but at the same time, globally, you are allowed to give it out based on doctors' requests. So all of that can happen in within a year from now.
Perfect. Just before we move on to the next question, I'm going to just create a poll at the bottom for the audience just to vote on. If you just, while we're doing the questions, please just have a look at that. The next question is from a gentleman by the name of Nelson, and his question is, why has something like this not been thought of until now? Is there any other company experimenting with nanocell technology and in this space?
Yeah,
That's a classic question, and we actually don't know, but we are the first people to have had the idea. Everyone, I have a saying, everyone likes to be first to be second. And so when nanoparticles started, synthetic nanoparticles and copolymers and all these sort of things started, everybody rushed in there. We are still the only people working on nanocells. And you know, it's terribly important because it has that immunotherapy stimulating activity. These other things don't, but everyone is over there.
Yeah, I mean, literally hundreds to close to several hundreds, I would say, synthetic nanoparticles. So it's a major area. But again, they're all struggling because no immune stimulation.
And they can't target them.
And an interesting thing about this point is that, is there anybody else doing any nanocell work? We have actually wrecked our brains trying to think, is there any other cellular structure in the world which can produce something like this, like a nanocell? So can you make it out of plants? Can you make it out of any other living system? The problem is, the answer is a straight no. You'd have to go straight back to bacteria because all those properties are found only on those bacterial cells, which is why all of this is happening. So even ourselves, if we wanted to make a nanocell from something else, we won't find it.
Okay, thank you very much for that. So Richard has posed a question that could be seen as quite similar, but it's, please explain the nature, size and measure of success of the human trials conducted, and are there others using the same technique? So the other technique, the question was referring to the other technique you've just addressed, but please look at the point of the nature, size and measure of success of the human trials.
So the classic result is from this pancreatic cancer trial. So currently we have cleared 17 patients. We only have to clear one more patient, and you know that phase 2A trial is completed. But 12 out of the 17 patients, which means 71%, have achieved stable disease and or tumor regression. Now at end stage in pancreatic cancer, it's unheard of. The answer would be 0. no patient would be expected to get stable disease or partial regression. We are over 70%, which is quite remarkable. Glioblastoma, same sort of results we were getting in the US trial, that almost every second patient was doing remarkably well when everyone's given up. Basically, they've just been told to go into palliative care and they're going to die within 10 weeks. Mesothelioma, similar result. So in 3 major cancers, totally unmet medical needs, we are miles ahead already, and which is why we already have enough data to now go to the US FDA for breakthrough designation.
And the 2 measures of success are people living a lot longer and people living healthily. So the quality of life has improved out of sight.
Yeah,
because
no
the immune stimulus too makes people feel better.
Yeah, virtually every technology out there, even the mildest of cancer treatments, is all still associated with a lot of toxicity. Whereas this thing, people are just coming to the hospital once a week, you get your jab, off they go, and go about their normal lives. Now, nobody would say that this is an end-stage cancer patient who is about to die, because, no sign of toxic side effects. Now, the people at Johns Hopkins Hospital, they say that even if you don't achieve any extra success in terms of anti-tumor efficacy, even then you're going to get on the market, because compared to everything else that's giving toxic side effects, if you have the option, what will I take? This thing will give me toxic side effect and I will live 3 months. EDV will give me no toxic side effects and I will still live 3 months. The answer is obvious. You'll go for something which is not going to give you toxic side effects. So it's a huge hurdle that is already crossed. Over 170 patients have been cleared in our clinical trials, and over 2,400 doses of EDVs have been given so far.
That's incredible. That really is a great statistic. So the next question from Nelson is, aren't you concerned about the giant chemo and cancer industry you're going against? How do you plan to avoid this brilliant tech from being acquired and put in the corner?
Very interesting point. And, in fact, that used to happen a lot. But, the world of business and commercial dealing became a lot more savvy. So, in fact, every licensing deal now is associated with one single clause, which says that if you do not progress this technology in a timely fashion along these milestones, then the technology comes back to us. Every biotech has got that clause. So in fact, no pharma is now capable of buying something and burying it. You just can't do it. Second point is, when big pharma are running out of their patents, and we have actually gone and said to them that, look, if you use your antibody via ADV, your patent life will increase. They said, no, we are not interested. If you've got something interesting, and if it supersedes ours, we will simply acquire it and dump our program. Because what the big pharma is looking at is revenue growth. So if something else is going to give them more revenue than their existing program, they're actually bearing their own programs. So against the chemo industry, etc., the pharma is not even concerned. They're only looking at what is the next growth phase, and they're acquiring them.
Fantastic. Thank you very much. So the next question is, how much does it cost to manufacture one dose, and what can patients expect to pay for the whole course? And I think that's on the basis of, you've got this great tech, but how accessible is it going to be for the average person that needs this technology?
Sure. Now, in fact, that is another one of these big boxes that we're ticking. So currently, all cancer treatments per patient per year, the cost, and the PBS is paying a big price for this thing. Every one of these cancer therapies is priced anywhere from $100,000 to $800,000 per patient per year.
Yeah.
Nothing is less than 100,000. Our single vial that we are just making here in the Lane Cove facility, it costs us around $400 for a human dose. I mean, it is staggering. The big pharma where we've gone to presenting our technology and the manufacturing, and they've sent out their manufacturing teams here to have a look at the whole thing. And they say, don't ever say that. Because they are so frightened about pricing.
Yeah.
So, but however, we feel that the big pharma will realize that with such a low cost of manufacturing, they can actually access the global market. So global market is much larger than simply focusing on the 7 rich countries. Instead of selling something for $800,000 to 5 people, but if you sell something at $50,000 to 10,000 people, you're going to make more money. So that's the rough idea of how the economics will work.
Fantastic. So Lawrence has got a question, and I think you have addressed it, but I'm just going to ask the question nonetheless, is can the therapy be used for all cancers, or are there certain cancers that have been excluded from your studies?
We actually believe it can be used for all cancers because we can change the antibody targeting to match a receptor. For example, liquid tumours like lymphoma, they have different sorts of markers on their surface, and we need to change the antibody. So we've got a cupboard full of appropriate antibodies we can use. We haven't done trials with lymphoma or leukemia, but we believe that they would work. It's just a matter of 30 people can't do everything. So yes, it's a one-stop shop for oncology, really.
Fantastic. So Richard has asked, what level of real interest has been shown by pharma companies, and do they see this as unique, defendable IP?
Oh yeah. They certainly see it as unique IP, and since COVID we haven't talked to too many pharma because we've all been shut down. But the big thing was, because we were first, they needed to see clinical data. That they've been out to various pharma, have looked at our manufacturing, and big vaccine makers believe they can, because we grow our bug in a big vat, and they can do that. It's more to see some clinical data, and as soon as we get an FDA approval to do this trial in the States, we'll be knocking on doors, and the banks will help us do that because they often have acquisition or deals done before an IPO, because there's a bit of a FOMO happening with the pharma.
Okay, fantastic. So just once again, for completion of questions, to address them, you have answered it, but it's what are the critical milestones and timing for phase 2 and 3 trials? So that has been answered in the previous last couple of minutes. I have a question from Rebecca. She's asked, can you utilize this new vaccine delivery for high affinity antibodies after you've had the current vaccines, Moderna, AstraZeneca, etc.?
Yes, interesting question. In fact, we thought of that as well, that you see, the moment this information comes out and our seminal publication is about to go to a top-level journal where this information will come out, that you do need high affinity antibodies. So can they reformulate the Pfizer Moderna vaccines to be able to make the high affinity? Is that?
No, I think the question is, if you've had the Pfizer or Moderna, right, can you have ours? Is that the question?
Yeah, so can you utilize this new vaccine delivery for high affinity antibodies after you've had the current vaccines?
Yes, absolutely. Yes, because it is going by that second pathway. So the person at the moment, say if you've taken a Pfizer vaccine, you'll get your low affinity antibodies. They'll be reasonably protective. It's not that they're not going to be protective. That's all fine. But in 6 months' time, the efficacy will almost go down to 0. And that's now pretty well known. So that's why they are all advocating for the third jab. Now, at the third jab, you could actually, if ours is approved, one could have our jab, and it will go by the second pathway and generate high affinity antibodies. So the person will be better protected.
So in our trial, we've actually gone back to the ethics committee to say, okay, we're going to include people who've been vaccinated 6 months ago because they have virtually no protection, frankly, at the moment.
Fantastic. Thank you.
Excuse me.
So we're heading to the last few questions. There's 4 left. I am aware of time, so just to let everybody that is attending, knowing that we are going to be recording this, so if you do need a hard deadline of 6, we are going to carry slightly over, The recording will be sent to everybody. So Lance has asked, the question is, how much has been raised of the required 25 million to date?
That's a tricky one. We're just, maybe eight, something like that. Because it's coming in bits. So we haven't added, I haven't heard from the CFO what the end of the week total will be, but I think about 8 or 9. Yeah.
And if we look at it, I mean, VentureCrowd's ability to invest only closes in October. So those numbers will only be finalized from our side, at least, at the end of the month. So thank you very much for giving us an estimation of where you're currently sitting against that target. Assuming you've raised around 150 million on the IPO, what will be the valuation of the company be?
Yeah. So as Alex pointed out, the median valuation at IPO at the moment is around the 460 to 500 million on Nasdaq. So post raise, but that already includes the 150 million raise in the IPO. So at IPO point, one would expect in the 500 million range. At the moment, the valuation is in the 180 million range. So therefore, one is looking at least a 3x step up at IPO from now.
Yeah, however, valuation is a funny thing. And if we are showing stellar results, like BioNTech originally was, if you just multiplied the number of shares by the share price, you wouldn't get anything like $50 billion. But, so it's all about our news flow too. So we might well be above that if our news flow is stellar.
Certainly, if we have a breakthrough designation in pancreatic cancer, that would most certainly trigger a pharma deal, because none of the pharma would then be in a position to say, oh, I won't make a deal, because if somebody else grabs it, which they would, and that's why we are also talking to most of the pharma, trickling out our results to most of the pharma, because what you have is auction economics at that point, where you want to buy, okay, who's the best bidder and who is the best player. So a breakthrough designation most certainly would push the valuation well past the 500 million mark.
Fantastic. That's amazing. So we've got a few more questions. Dan has asked, are there any known side effects, and risk associated with your tech?
So I think we've de-risked it clinically very much so. The side effects are about 50% of people have a slight increase in temperature, and sometimes they get flu-like symptoms, which is not entirely a bad thing. That means your immune system is getting kick-started. So that's all we're seeing. We are not seeing any damage to other organs or white blood cells. They actually go up because they're immune cells, and we're increasing those. We don't see any blood work that is dangerous, and everyone feels better. So our safety profile after 1,000 doses is really good.
And one more point to add to that is, many of our patients have received anywhere from 15 to 70 repeat doses. Now, if there was any possibility of any toxic side effects, you can imagine, is there any cancer treatment that you can literally give 70 times to a single person and have no toxic side effects? So this is what we are seeing. And we are not terribly surprised that this is a very, very safe particle, simply because it's completely inert until it goes into the cancer cell and dumps only in the cancer cell. It does not go into any of our normal cells. Now, if you even look at the COVID vaccines, like these lipid nanoparticle vaccines, when you inject it in the arm, a lot of the cells in your normal cells in the arm, they will also take up the vaccine. Now, getting into normal cells is where all these risks start. But if you're not getting into any of the normal cells, then this is remarkably safe.
So you just inadvertently answered the next question from Richard. His question was, how many doses in a treatment regime for somebody? Does that depend on the cancer? Is there a set number that you're working with?
at this early stage where we're in very sick people, there's no set number. We treat them for as long as they want to be on the trial, or for as long as it is showing benefit. So that's why, you know, somebody who's very sick might need 70 doses. But as we go up the line and come behind first line, those people will be much better. They won't be as sick. And we think they will require less EDVs to get their system going.
So we have clear examples of those, like glioblastoma, mesothelioma, where with one cycle of treatment, we almost got a person into complete remission. So that's what would happen when it goes up the line. So one would expect that this sort of a treatment, say once a week for 8 weeks, would be as much as one would need. And then you monitor every 6 months. If the tumor came back, then you can easily retreat and you can knock the tumor out. So the lowest would be at least about 8 weeks of treatment, that is 1 dose per week for 8 weeks. And the longest one could remain on the study because they were too far gone, and it takes a while to build up the body back to health. You could a person, we have people who have gone for a whole year.
Thank you very much for that answer. So we have 3 left, and we're not going to be taking any more questions after this, just in the interest of time. Nelson has asked, as pre-IPO investors, will you get privileged access to the drug? Will the investors get access to the drug as a pre-IPO investor?
So the rules and regulations are not based on that, because you see, from a company standpoint, we have to follow all the rules and regulations from the TGA, from the FDA, etc. But what there is compassionate access, where anyone can access it. So we just have a TGA notification. So we notify the TGA, and so long as the oncologist says, I think that what I'm about to do in this patient, this EDV is likely to do better, so I want the EDV. Then before product on the market, you are allowed, through investigator-related request, you can give to anyone in the world.
Fantastic.
I should just point out that Carolyn, our pancreatic trial is called the Carolyn trial. Carolyn came down with pancreatic cancer, and she was our very first investor. So maybe investors, have the inside running because they know what we're doing.
Yeah, well, in her case, she was 2 days away from death. And we actually took the wire to the hospital, first dose, and the oncologist called us on the side, and he said, look, do you really want to do it? Maybe in a day she'll be dead. We said, this woman was the very first person who gave us $2 to do our research. And are we going to walk away? No. And we went ahead, and we did remarkably well. So investors, of course, you help us. We never turn, we don't turn our back on, even on animals. So nothing to worry about that.
You've just reconfirmed why there's such a business alignment between what VentureCrowd is looking for in businesses and what you've brought to the market. So it's very much a space of finding and funding futures, and you're giving people their futures back through this technology. Richard has asked, and it's the second last question, is what runway does 25 million provide you?
We're running at about 600,000 a month. We'd like to actually ramp that up, and it will ramp up as we do more trials. So that'll take us a long way.
So one's looking at least early 2023.
Yeah.
Minimum.
The last question is from TJ. For this raise, round C, is the price per share the same for all parties taking part, or are institutional investors getting a different price per share?
No, all the same. Even we are put into this round as well.
Yeah. All at the same price.
Yeah.
Well, doctors, that's the end of the Q&A for now. For those investors that are still with us, please, and if you haven't yet, please just have a look at the poll that is running at the bottom of the webinar and complete those. So I'm going to just close out the webinar. Thank you for those that have managed to stay on. I know we have run over a bit. Alex, I'm just going to hand over to you just to round up some of the salient points and just to surmise the the fireside chat that we've had with Dr. Jennifer and Dr. Himanshu, and then we can close out the webinar.
Yeah, thank you, Tam. I appreciate it. I mean, we've certainly gone through a lot tonight, and I really appreciate the questions coming through. It really shows that audience members are really thinking about this thoughtfully and really committed to seeing where this drug can get to in its commercialization. So just in terms of what we did talk about tonight, we covered off EDV, which is Engenic Dream Vector, the technology that is behind Engenic. How it's protected by its extensive patent portfolio in each of its different verticals, from manufacturing to clinical science and everything in between that creates the business. We also talked about the team behind Jennifer and Himanshu that is leading towards their funding round for this pre-IPO. So again, this offer is open on VentureCrowd at the moment. It is a $159 million US pre-money valuation. We will only be keeping it open to the 31st of October. So if you have any questions following this webinar, please revisit this webinar when you receive the recording and reach out to our team so we can tell you more about it. Before we do go, I do want to just ask one question to Dr. Jennifer and Dr. Himanshu, and that is, how will the world be a better place thanks to Engenic?
Well, it'll be a better place because our heart's in the right place. When, as I said previously, we're not just looking to become rich and famous. We want this technology out to the people that deserve it most, some of whom have intractable cancers. And for example, in pancreatic cancer, there have been no new drugs in 30 years. And the ones they've got are hopeless. Also, we're very interested in developing countries because the cost of goods, the fact that it can be stored at room temperature and you don't have to have a cold chain, etc., make it very desirable for developing countries. And we do have some interest from places like Kenya and India and China. So that's good.
Yeah, and I think that finally I would just simply add that, you know, when you look at a cancer patient who comes at the end stage and the complete loss of hope, because even the oncologist says, look, I've got nothing for you. It's it. You go home. And like that death sentence while you're still alive, you know, is we should never have that. And this technology will change that. That nobody is ever going to think that, look, I've been diagnosed with cancer and my story is over. No, your story is not over. And you're going to live on and you're going to be happy. You're going to go back to your family and you can have more dogs and cats as well. That's it.
Well, we certainly are excited to support you in these endeavors and the outcomes that it can have for the entire global economy and humanity. So we do appreciate your time tonight.
Okay,
Thank you
very much.
Thank you very much, Alex and Dr. Jennifer and Dr. Himanshu, for all and to those that attended. I hope that the information that was provided you found informative, helpful, and gave you some sense of hope and idea of what this business is about, what the tech is about, and the passion that is behind it. So on that note, I just want to let those on this webinar that are still with us know that if you have any questions, you are able to contact the team on a contact number of 1300 039 655. Or alternative, if you have questions and you don't have the time to make the phone call, please just pop it in an email to the following email address, which is investor@venturecrowd.com.au. Thank you so very much to both of you and to Alex for your time. I know we have run over. It has been absolutely amazing. It's given me goosebumps. It's gotten me excited, and hopefully it's done the same thing to those on here. I hope that everybody has a really good evening and a safe evening, and the webinar recording will be sent out in the next couple of days. Thank you, everybody, and I hope you have a lovely evening.
Thank you.
Bye.